welcome back to yet another Journal Club brought to you by Lifespan.io. I'm Oliver Medvedik, along with our cast of regulars here. We have yet another longevity-related journal article for everyone. This one was written up, I believe, on our blog. So if you want the condensed version and all the, without all the figures that we're going to go through, do check out our website. So without much further ado, let's share our screens here, and let's take a look what we're going to be talking about. What are we talking about? Oh, this paper. Okay, whoops. Oh, my computer is acting up. You know, when you're on a laptop, right? So sometimes it senses your finger and it's not there. And anyway, things just move around. Nature Biomedical Engineering. So we have this paper, this article on intradermally delivered mRNA-Encapsulating extracellular vesicles for collagen replacement therapy. I don't know a whole lot about extracellular, these little vesicles. So cells tend to shed. People have been studying these for not a whole long period of time, but eukaryotic cells shed these vesicles that are either endosomally derived or shed from the plasma membrane, and they tend to encapsulate a whole bunch of biologically active molecules, such as, I believe, both coding RNA, such as in this case mRNA, but also non-coding RNA, and they are, they believed to play a role in signaling and other events within metazoans. And this paper here, what they are doing is basically generating these vesicles to package mRNA and using them as a delivery tool for collagen replacement therapy. So they are encoding the mRNA for, I believe it's Col1A1, encoding collagen, and using these vesicles to essentially fuse with cells and deliver that cargo. So essentially the cells can now generate collagen. So cells that have basically ceased generating collagen. Why are they using these vesicles instead of other delivery methods? Well, the authors claim that this is a superior technology. You get better delivery than using needles, for example, which they look at in this paper. And you also get better responses compared to another small vesicle mediated delivery system, which is lipid nanoparticles. And I think we've heard these as a means for delivery for the current line of COVID vaccines that are mRNA basically encapsulated within these lipid nanoparticles. The authors claim that some of these nanoparticles could have allergic effects, could promote inflammation. So there's other problems, and you don't see these problems with these kind of more naturally derived extracellular vesicles, or EVs as they call them. So this paper has a number of authors. So it's EU et al. And basically it's a collaborative venture between multiple schools, basically in China and also here in the US. So it's the University of Texas, also Vanderbilt University in Nashville, Tennessee, and the Peking University Shenzhen Graduate School in China. Right, stop sharing. I thought we changed to Beijing. Well, that's a little, right? Is it now, or is it now, or is it just Peking University because of tradition? It's still Peking University. Anyway, okay. So let's go back to the paper. So what do they do? So there's a whole bunch of figures here. I'm going to move this to the side here. So the first thing they do is they generate these extracellular vesicles using a novel technique. So there's another bit of technology that they employ, which is the cellular nanoporation, right? So the way you basically, so they use a plasmid to get into cells, can't remember which cells they used here. They test the first figure they're testing. So they're going to later do all of this in mice, but the first figure they're using it in human cells. So these are neonatal human fibroblasts, and I think they're basically generating these vesicles also in neonatal fibroblasts. So they're getting these plasmids in. So this is a summary of the technique here. They're electroporating them. So electroporation is just a technology where you zap cells and basically use the potential to drive the plasmids into the cells, and then they express messenger RNA, and then they start shedding these extracellular vesicles. And I'm not entirely sure how what would prompt. Oh, Micah, yes.
So I would assume that neonatal human dermal fibroblasts would already have tons of collagen expression in them. Why do they need to add more?
It's a good question. What they do is, what do they do here? That's a good question. The later experiments, they use, I think, the younger mice too, and they use photoaging. So they basically zap them probably with ultraviolet light. Let's see.
I didn't see anything in the paper that mentioned why they did this. It just seems odd. Like they're using a cell that seems like it would naturally be extremely high in collagen.
Yeah.
Which seems like a reasonable choice, right? And then they go and add more collagen. And I just didn't understand why they added more. Is there not enough? They just upregulate so you can get even more of it out? Is it worth the effort?
Yeah, I think. Well, I mean, let's see. For the mice here, so they're using these to so this system here, they're generating. So I think they're using these as a factory to produce these. I think they're using throughout the paper these cells here as a factory to produce the nanoparticles.
Yeah, so I agree. That's my understanding as well. It just seems like those particular cells would produce these EV nanoparticles on their own, without any coaxing. You just simply go to them and dish now to assume that they just kind of naturally shed collagen containing EVs. Maybe I'm wrong there, and maybe that's the issue. But if these don't naturally produce collagen EVs, then could you do the same thing in literally any cell type, and it was just purely coincidental that they happened to pick skin cells?
Well, it's a good question that you raise, because later in the paper, they show that they do get effects using extracellular vesicles that aren't loaded. You get a much more robust response with these collagen 1A1 EVs, where they're these EV number per cell. So after control, then you have this is bulk electroporation, so just using traditional electroporation, and this new technique that they call I think cellular nanoporation, where monolayer. So you get a lot more.
I see. Yeah, I totally missed that graph right there that you pointed out. I think that answers it. So if you just use dermal fibroblasts, you do get some
a little bit,
EVs out. But with this technique, we're basically mass producing, and that's kind of suggests to me that you could probably get the same effects in any cell type.
Yeah,
just they happen to use these ones because it's for whatever reason.
Right, but my question that but you do raise a good point there, because you do get an effect with these. I mean, I think again. Papers always generate, at least, I don't think there's a single paper that we've gone over or I've ever read that hasn't generated additional questions. That's just the way science works. We'll get to that point, but they do see effects with increased I guess less wrinkling and so on and so forth, if they just basically treat the mice with these EVs that haven't been loaded, or that haven't been derived from electroporated cells that have had plasmids. So there is something else packaged in the extracellular vesicles, or maybe it's the vesicles themselves. I don't know. That's basically can trigger some sort of beneficial response. And what that is, it's kind of hard to say, because I think they're very heterogeneous. So again, going back to how they generate these extracellular vesicles, so they have EV number per cell. So that's A is the setup. And then if I go to B, produced by untreated HDF-PBS as control BEP or CMP with okay. So you get so just this electroporation event generates these vesicles, right? So this is like not with a plasmid. So basically, if you zap them, you get this response. And I'm not sure what this what the physiological basis is for this, right? I mean, because it's pretty dramatic. So I'm not really sure what the why that's the case. And then they basically, go on to characterize these vesicles, which they show that the size, ranges in this range. You get peaks at about 100 nanometers, right? So 0.1 microns, and pretty much everything is between 50 and 450 nanometers. So, smaller than bacteria to about the size of a really, really, really tiny bacteria. So that's kind of the range of these things. Okay, so then I believe in D they do the loading. So so when they do the electroporation with plasmids, you have more of, so they do RT-PCR on these vesicles that they isolate, and they show that more of them are loaded with this mRNA that's COL1A1, so encoding collagen, using this cellular nanoporation technology. And then they show that you have expression. Well, actually, no, that's just RNA. Let's see. Did they look at expression of protein? No, gel expression of total RNA extracted. So I believe that the size of the RNA that encodes the collagen is about 4,000 nucleotides. So so there's some degradation here with this synthetic mRNA that they have. I believe that's a loading control. I don't know why that's a breakdown of that, but that is a control that's not loaded. So a whole bunch of tiny RNAs. I don't know why there's kind of nothing down here. I would expect to see kind of maybe a bit dirtier if this is actually isolated from the nanoparticles. Total RNA extracted. Yep. So it's kind of interesting that you got this 4,000 nucleotide, which is corresponding to the mRNA from the plasmid that's encoding collagen, but I'm not sure why there's no stuff down here, but it is in the control. So the control, I'm assuming, is basically just electroporated, but with no plasmid. So next, F is kind of interesting to me. So the proliferation of neonatal human dermal fibroblasts after treatment with different protein concentrations of different protein concentrations. Maybe that's a typo. You determine the concentration of extracellular vesicles by protein. I mean, they are vesicles, so they'll probably have protein embedded on their membranes, but EV concentration micrograms per mil, I guess you do. So they don't mention this here, but when you, I guess, subject the dermal fibroblasts to different concentrations of these these EVs, these extracellular vesicles, the proliferation goes up by quite a bit. If we're looking at 100%, or day zero, right? Is this day zero? No, that's a concentration. So let's see. How much? 48. Okay, here's the time. Yeah. 24 hours and 48 hours. I'm not sure why EVs would be promoting proliferation of neonatal human dermal fibroblasts. And if anybody wants to comment on that, they don't mention that in the paper. They do a proliferation assay. And these EVs are loaded with a whole bunch of things, I'm assuming not just the collagen that they're expressing, but probably some other stuff as well. But let's assume it is just the collagen that they're expressing. Would the collagen itself have a stimulatory effect on neonatal human dermal fibroblasts? Does anybody know? I'm not sure. Okay, silence. Everybody else is like,
I do not know.
Yeah. So yeah, I'm curious now. And sort of a corollary to that, if I were to put these EVs not loaded with COL1A1, would it also have a proliferative effect on neonatal human dermal fibroblasts? I don't know either. But yet it does, at least with the COL1A1 EVs. And I'm not sure about the significance of that. Yeah, I didn't see that. It was mentioned. Maybe I missed it. It was, I read it a while ago, but it was mentioned here. But it wasn't, that yes, it happens, but not why. Maybe it's just assumed that we expect that to happen. Proliferation of fibroblasts was observed to increase with COL1A1 EV treatment in a dose-dependent manner. And that's that. Okay. So now we go into photoaging models with mice. So now they're switching gears and using these vesicles and looking basically, looking at mice that have been photodermally aged. So I'm not sure. I don't think these are old mice. I think these are young-ish mice. Yeah.
Couple comments before we move on from the setup. I don't know if anyone has the answer here, but I figure I'll throw them out there. Could we use iPSCs instead of neonatal cells? It seems like from a treatment standpoint, long-term, more viable than trying to acquire large volumes of neonatal human dermal fibroblasts.
Yeah, good question. I haven't had a chance to read a review paper. First thing I do is just dive into a review paper on these extracellular vesicles and see what are all the platforms used to generate them. I would be surprised if human dermal fibroblasts neonatal are the only source of. I think they're all over the place. I think like basically every cell is doing it. And if I were to guess, I would think that depending on the cell type, they're most likely loaded with different cargo, and they probably have different functions. I skimmed briefly one review paper, and it talked about as means of signaling by delivering non-coding RNAs. But I didn't really have a chance to kind of go through it much thoroughly. So yeah, I don't think there's a real solid reason for not switching to what you just suggested.
I mean, it might just be that this was simpler. This is what they had around in the lab at the time, and so they just used it.
It could be, and they were growing like crazy, and they've already used this in there because they cite their previous papers where they developed this cellular nanoporation that they use in this paper. So it could very well be that they already had this up and running from their previous paper, and it was just easy to basically segue into this.
Yeah. The other thing I was curious about is, do we have the technology yet to generate these EVs with yeast or something? Do we have the engineering chops to do this, or do we still don't know enough to be able to produce these ourselves? It seems like if you're trying to mass produce these for a therapy, it seems like ideally you just have a yeast vat that is just making these and spitting them out rather than trying to induce human cells to do the same thing.
I don't know if yeast actually do produce EVs. That's a good question. I mean, they're surrounded by a tough cell wall.
They may not. The question is, can we engineer them to do so? We engineer yeast these days to do all sorts of things they didn't originally do. I'm wondering if this is, one of these things we could move over to yeast instead of human cells. And if so, is there any advantage to that other than just we have a little more control? They're less complicated. You're less likely to get something else in there.
Yeah, I think they're one of the main reasons they use these is because it doesn't promote an immunologic response to the host, because they're going to be using this as a therapeutic to inject into people. So, or so I would think that if the thing is loaded with a bunch of yeast proteins, you might have an inflammatory response, and it might be more difficult to engineer a humanized yeast. I'm sure, given the time, money, and resources, we can do such a thing, or we can just use human cells, right? So yeah.
Yeah, that's fair. That's fair.
So that might be the reason why, just technical limitations, and that it's eventually going to be used as a delivery system in humans as well. But to your point, yeah, you can probably get to really minimize any kind of response, because I would assume if these things are generated from plasma membrane, they're probably coated with M8C proteins. So if you're going to be injecting them into different people, try to get these things as immunologically matched as possible. So with your, they talk about one of the reasons they're doing this is it promotes less of an immune response. But I think these are immunocompromised mice. I'm not 100% sure, because I think they're using these human vesicles here. So if you're going to be doing this in humans, then you clearly have to have other proteins that might trigger the immune response, such as the M8C class proteins, the major histocompatibility complex to be matched. But to your point, Micah, yeah, if you use iPSC cells that are derived from a patient, then you should be able to generate like completely matching vesicles that are, or you could probably modify them so they, some cell line that doesn't produce M8C proteins and have it sort of generic. Anyway,
I think there's people working on that problem, universal cell lines for that specific.
Yes. So I think there's a bunch of workarounds that could be done for that, and then everything else, the phospholipids, right, are matching. So there's no weird funky phospholipids that can trigger in immunologic response. So if they use this as a delivery method, then it's gonna be recognized by the body as being. But yeah, it's a fascinating subject. I gotta dive into EVs a little bit more to see what are all the different things that they're capable of, and and are there specialized receptors that bind to some EVs and not other EVs? Is there a significance to the size of all these different extracellular vesicles? Right? Is it the more you dive into molecular bio and cell biology, the more you see that what looks from a distance as kind of randomness is actually not random. There's an organization there. So it's not just all helter skelter like these images show. There is a pattern there, which breaks down as we age. Okay. So, in vivo kinetics of COL1A1 EV mRNA delivery and protein formation in murine skin. So now they're delivering these vesicles, using intradermal injection and saline controls, and then they're measuring different time points. Half a day, 1 day, 2 days, 10 days, 14 days after injection, and then they're visualizing the RNA in, I believe, skin cross sections. And also the COL1A1 EV-derived protein that's being expressed. And there, I don't know if it's probably in the same plasmid, which is an expression of GFP, just to show that, for easy visualization of where it's being expressed, although it seems like they're capable of staining also for COL1A1 EVs. So what they see here is that, it's a transient expression, right? So we have control up here, which is just saline. I think these are photodermally aged mice. So let me take a look at here. Oops, this is already figure 5.
Yeah, UV exposure for 8 hours. Something.
Yeah. So you could try this yourself. If anybody ever goes to like a skin tanning salon where you sit under those UV bulbs, your skin turns into kind of like a parchment-like quality, right? If you sit under there too long, so it's kind of a similar effect. So it's not good for your skin. So you want to simulate that yourself, go right ahead. So we have a control up here, right? And then we have staining for mRNA using this staining technique that they mentioned below, COL1A1 extracellular vesicles, intradermally injected. And yeah, you can see the levels go down dramatically after about, 2 days or so, 3 days. So 7 days looks about equivalent to controls. And here they have the average number of dots per cell, which are basically staining, probably similar to probably hybridization with a staining complementary mRNA. So average number of dots per cell. So you can see it drops off precipitously after about 48 hours. That's the control. And then they have GFP intensity, so the proteins obviously will linger a little bit longer. So what do we see here? This is staining for proteins. So again, cross sections, DAPI, GFP, COL1A1, merge, and let's see what figure is this? C. That is C. And C is immunofluorescence over time of COL1A1 EV-derived protein by visualization. Co-localized GFP and protein. Let's see. E is just quantification of GFP positive cells confirms that EV-derived protein grafts decrease in a time-dependent manner. I don't think is there a comparable curve for the collagen itself? I'm not sure. So GFP expression actually starts going. It's kind of interesting that there's this delay. It says time D 3 days, GFP positive cells. Is that because they're dividing, or is that, expression does lag, of course, behind, or translation lags behind expression, but not that much, right? 3 days, and
maybe
peaking about a week.
Maybe it's the time it takes for the EVs to actually get into the cell and then release their cargo. Maybe that's a slow process.
I don't know what I would. Yeah, I don't. There's not a figure that talks about the kinetics of that. I would assume that it's pretty darn fast, just because.
Yeah, same.
Just because the. Those are the EVs. So those are the dots. Those are probably the extracellular vesicles. Let's see, DAPI merge. It's hard to say if this is inside the cell or kind of co-localized with the cells, right? It's not really. If the mRNA is in there or out there, I would assume, again, I have to do some more background on the dynamics of all of these EVs, but I would assume that it's pretty rapid, because if it's acting any in the same kind of kinetics as or similar to lipid nanoparticles, right? When you get it injected, for a vaccine that utilizes those, it's pretty instantaneous, I would think, that you get this fusion happening with the cells at the site of injection. But maybe it's something different with EVs. Okay. All right. So now we have images of mice in flesh. So naked mice. Here we go. So COL1A1 EV mRNA delivery reduces dermal wrinkles in a UV irradiation photoaging model. So we have a couple of controls here. So sham is basically not UV irradiated. Saline is just injection with saline solution. So if you go out into a sun tanning booth, this is what happens. So try to avoid those. So we could see with the sham, these mice have been kept away from harsh UV, and look how smooth that looks, right? And now we have saline, which, so after days, day 28, right, still has wrinkles. I would have thought that it sort of would have recovered fully by day 28. It's pretty harsh photoaging taking place. RA is retinoic acid, which I believe is used as a topical treatment to basically slough off, I believe, and older cells on the dermis and promote growth. Unloaded EVs is basically extracellular vesicles without the expression of the mRNAs. COL1A1 lipid nanoparticles. I don't know how they generated that, but that's basically. Hold on a second, I have to mute myself. There's noises in the background. Oops. Sorry about that, folks. Okay. And COL1A1 lipid nanoparticles. And then finally, we have the COL1A1 extracellular vesicles. And you can see down here just by eye that there's a, it's an improvement over time. So let's see. So here is total wrinkle number and total wrinkle area that they, not sure how they measured it, probably put a box around it and had some software draw in the wrinkles and calculate them, right? So here, all sorts of sounds when you live in the city. That's ambulance, let it pass. Okay, so those are the, that's the data here, and this is what I mentioned before that was interesting, where if you have unloaded EVs, which is this black triangle here, right, you do have a decrease in wrinkle number. So saline is no change, so you're getting this effect with just unloaded extracellular vesicles. Yeah, Micah?
So my theory with the unloaded extracellular vesicles is maybe it's just the way they filtered. So they're using these human dermal fibroblasts to get the extracellular vesicles. And if they're just kind of assuming, okay, we didn't, use our magic stuff to make them produce massive amounts of the collagen ones, and they just picked whatever, then some of those are still going to have collagen in them because the human dermal fibroblasts are generating collagen. So maybe they just got a little bit in there, kind of on accident, not intentional.
Yeah, that's likely. We could again, it'd be interesting to do some additional experiments, like have human dermal fibroblasts or other cells, or or knock out the collagen production, right? And basically make sure that, okay, maybe it's a collagen. And then if we do that and we still see the same effect, then it's like, oh wow, it's not the collagen, it's something else. Yeah. So they noted that and they mentioned it in the paper. And I don't think they go much further than mentioning that. So there's an area of exploration there, and then of course with the Col1A1 EVs, right, you get a much more dramatic effect here. A little bit, a bit of a strong effect also with the lipid nanoparticles, but the most dramatic effect being with their Col1A EVs after 28 days decrease, better than all the others, right? And this is total wrinkle area, so you basically get this similar response that tracks, I believe, I didn't get it too carefully, but I believe it tracks or trend, the trends that we see up there. So that is with an intradermal injection. So they mentioned what they use of a 28 gauge needle and inject maybe multiple sites. So then they do basically now some histology where they take those mice, they sacrifice them, and they do basically tissue cross sections, and they do staining. So in C, they basically stain for collagen using this Masson trichome collagen staining. So basically, you'll have a thicker dermis, right? If you have more collagen being expressed. So basically, dermal thickness goes up as well, right, with the EVs, and there's a little bit of effect with these unloaded EVs, but more dramatic effect with the extracellular vesicles. And now they also look at, I believe, they look at protein expression of collagen 1A1, and you get better or more intensity over a greater area using these extracellular vesicles. So that tells me that it's probably getting into more cells also than the lipid nanoparticles as well. Interestingly, yeah, you might be right, Mike, about the collagen being kind of sneaking in, right? Because, well, I don't know if this is significant, but they'd have to do more mice there to see if we have a significant effect. But it could be that that it's stimulating. Well, it could also be that it's stimulating collagen production indirectly rather than delivering the collagen expression. But anyway, so now they switch to another technology that they've been developing. So they were developing, the production of extracellular vesicles loaded with the mRNA, and then they also, developed a technique to generate them, in bulk using this cellular nanoporation technology. And now they're coming up with this new delivery technique to basically get it into this dermis of the mice using these microneedles. which I believe these microneedles are manufactured using hyaluronic acid. So I didn't know you can make hyaluronic acid into kind of like a stiff enough solid to make a little needle, but evidently you can. And here's their production facility using these PDMS molds that are vacuum pumped with slurry of hyaluronic acid that's mixed with the extracellular vesicles. So they're embedded in this, and they make basically this little dermal patch that's got all these little needles. They do also some analysis that they mentioned in the paper, discussing strength of these tips, right? They do some, engineering calculations to show that they're strong enough to puncture into the dermis, right? So they don't go very deep, right? They go into the, so you could probably hardly feel that. It's like a little, little Velcro-like patch with a whole bunch of little needles that over time, very brief time, right? So 15 minutes, they start to dissolve and then release their cargo. And they use, so the first thing they do is they basically just test them out and see how they function. And they use this di-I labeled EVs, which is this dialkylated, it's basically this lipophilic dye that basically fluoresces, and it's used as a way to track where things go. So it's kind of a generic fluorescent dye, and they're basically just using that to see how well these extracellular vesicles that are loaded with this dye get to an area. And here is so they compare it to intradermal injection and their hyaluronic acid EV system. I don't know what size hyaluronic acid they use or where they got the hyaluronic acid. It's probably somewhere in the materials and methods, right? Because there are different sizes of hyaluronic acid, and some of it can promote an inflammatory response, and some of it is beneficial, right? So we all talked about in the past about the naked mole rat, and it's little mammal that basically lives really, really long and has I don't think it gets cancer, but it secretes hyaluronic acid, like a lot of it, and it's extra big, so it's a very long chain hyaluronic acid. And hyaluronic acid is basically found in your joints, right? So it's there for cushioning, but probably and a lot of other roles in your extracellular matrix. So I'm not sure exactly. We could dig around in this paper, but see what size HA they used and where they derived it from. But anyway, that's their delivery technique here. Okay, and they show that it has a good distribution, better than an intradermal injection, which you kind of expect, right? Because you've got a whole bunch of these little microneedles spread over an area versus I think they use 5 needles that are injected. Plus, if you're doing intradermal injection, it's probably a lot of variation with the operator, how deep you inject the needle, and so it's probably going to be not as consistent as those patches. So here's normalized fluorescence and intradermal injection, so it lasts a bit longer. I would probably attribute that to greater number of EVs being distributed over more cells. So that's their microneedle system. Now they do experiments with it. So now they basically repeat what they did before, but using loaded and unloaded extracellular vesicles, using their microdermal hyaluronic acid dissolving needle technology, and comparing it to needle injection. So they've got a control with, so these are photodermally aged mice. So you want to see some skin? Here's some skin. Here's mice. So saline up here. A lot longer duration of experiments they did in this case here, all the way to day 91. So needle injection, hyaluronic acid, microneedles without anything, unloaded EVs with loaded onto the mixed with the microneedles, the HA microneedles, and the Col1A extracellular vesicles loaded. Can you already see down here? It looks like it's doing much better after day 7 with these with these Col-1A collagen loaded microneedles. So total wrinkle number, total wrinkle area, saline up here, the yellow, control, and you could see that Col-1A1, the black line down here, you get a dramatic effect. The wrinkling does, come back, the total wrinkle number over time. And again, I'm not sure exactly how badly these mice were. I mean, it looks pretty badly. They've been photodermally aged. They eventually the wrinkles come back. So yeah, it's you could probably design some experiments where you have different ranges of photodermal aging and probably some other types of aging as well. But anyway, that's their model. So you do get a dramatic response. Kind of much shorter lasting with the needle injection. Probably because they're looking over an area. I would assume, you probably get more inconsistent results with the needle injection. And again, unloaded EV, you get some effect there. Let's see, HA. Interestingly, you get some effect with just the hyaluronic acid microneedle. Again, the hyaluronic acid could be doing some beneficial. I mean, it's very, it's significant. It's this is total wrinkle area, not so much here, but hyaluronic acid is a biologically active molecule. It is found in your extracellular matrix. Provides cushioning to joints, also holds lots of other cellular factors in place. So it it might be. I don't know if they mentioned that in the text as to
the saline control here. I don't think the saline control is with the microneedle. It's possible the microneedle itself, like the act of kind of puncturing the top layer of the skin, just had some effects, like triggered wound healing or something.
Yeah, it could be. Yeah, it could be. It could be. The saline is not a. Well, actually, saline was that?
I think they're doing the needle injection saline here. But
well, because they mentioned, actually, good question, because they have the blue as separate as needle injection. So yeah. Well, actually, the needle injection would be, I think, with the Col-1A1 EV. So saline, yeah. Let's see, did they put saline in a needle, or did they put saline just? I don't think they. Yeah.
I guess the microneedles are made out of the acid, right? And so I suppose there is no control of puncturing without the HA. Is that correct?
Well, it's just basically HA, just HA, I think, with nothing loaded in it.
Right.
And then the only control is the only other control is having HA loaded with unloaded EV vesicles. So yeah, it could So it could just be that just HA alone, or I mean, you could test for this by just basically generating a microneedle that's not made of HA, just like plastic, like polyethylene or something inert, something that's going to promote an immunologic response, and basically see what happens, and whether it's just a I mean, because you puncture the surface there, you get cells releasing histamine, all kinds of immunologic responses, so that could certainly do it. Okay, but bottom line is you get much more beneficial effects with this that last for not forever, but for like 2 months or so. Okay, so what's the last thing they do here? So one more experiment. So they do basically again. Sacrifice the mice and do dermal histologic analysis to show dermal thickness. So Mason trichome staining to show that you get increased dermal thickness and collagen expression. With these mice, 1 month, 2 months, 3 months. Saline needle injection, HA microneedle, unloaded EV with HA microneedles and Col-1A EV microneedles, and then dermal thickness after 1 month, much more thicker with the Col-1A, significantly thicker, and then it kind of starts to drop off, and still slightly higher than the saline after 3 months. I don't know if that's significant. And additional experiments with basically expression of probably won't go into, just the expression of COL1A plus GFP in the areas of injection, which basically last longer with the microneedles. So I'm going to stop sharing here. So that's it. So that's the experiment. So you can certainly get less wrinkling by expression of collagen and having it delivered using these vesicles, using these variety of approaches. And Micah, yeah, you got a question?
Some comments. So the paper I found very interesting for the EVs and the microneedles, just in terms of delivery of therapies, to the skin in particular. The collagen aspect was surprising to me because I feel like they didn't actually cure the age. Like early on in the paper, they make the comment they say that the UV causes or down here. The paper claims that UV exposure destroys collagen in the cells and the ECM. But this must not be the problem, because if that was the problem, then restoring the collagen should have long-lasting effects. It should not be transient. And the problem is this paper showed that these effects were very pronounced. Like, we took the mice's skin back to youthful or undamaged place, and then the wrinkles came back. But the wrinkles, these weren't naturally occurring wrinkles. These were wrinkles because of the UV damage, which means the damage that the UV caused that was leading to wrinkles is still there. Like, we didn't cure the underlying thing, because otherwise it wouldn't have come back. And so that suggests to me that this is, at best a temporary makeup lipstick on a pig, so to speak. Aging is not cured here. All they did was address the symptom of UV exposure, and that was kind of disappointing, because it felt like the paper felt directly contradictory in that sense, in that they state UV exposure causes these things, and then we fix those things, but it didn't actually cure the problem long term.
Well, at a deeper level, I mean, the UV is gonna cross-link also your DNA, right? So you're gonna have somatic mutations as well.
Right.
And I don't know how much of that is been repaired, at least under the harsh conditions that they did here, and whether or not you're also getting aberrant collagen and other aberrant proteins being produced as a consequence of this photoaging experiment. That keep going on after the mRNAs have disappeared here.
Yeah, it's interesting, because if you assume the UV exposure damages the cells, right, like just due to somatic mutations, like you said, then why does restoring the collagen fix the wrinkles right away? Is it that the UV damaged the cells, specifically the collagen producing part of the cells, but the rest of the cells were still able to continue to survive? Because the UV wasn't so extreme that these cells died. The cells still survived, but they just didn't produce collagen anymore. It just feels weird to me. I don't have an answer for it. I don't have an explanation. It just, something doesn't add up in my head, and I can't figure out what that is.
Yeah, John?
Yeah, my guess as an answer to Micah's question is that it was mRNA that was delivered, not DNA. So we wouldn't expect it to be long-lasting. That's my guess.
Yeah, so I definitely agree with that. And that, I had another comment that I wonder if you could use EVs to deliver DNA instead. I don't know if
you could, probably.
Limitation there. But even then, the thing that really bugs me about this is that I expect UV damage to be kind of just stochastic across your DNA, right? It's going to randomly destroy things. Why is it that these cells specifically seem to stop producing collagen, and they need this help?
It's stochastic. I think thymine residues are the ones that get cross-linked. I'm not sure. I think there's probably I would assume that probably for that reason, and probably other reasons as well, that you get a stochasticity, but it's probably constrained to certain regions of DNA, probably not randomly, not purely across the board. And I don't know where the collagen gene is particularly situated. Also, if these cells are actively producing particular proteins, I would think that those genes are probably in a much more precarious position while they're being actively transcribed, so they might be much more easily damaged. I don't know. That's a good question. That's actually a very fundamental question. I have to take a look at some old papers and see if somebody's done such a basic experiment, which is like, are we going to blast cells with a whole bunch of different mutagens and see under which conditions and which cell types, which genetic regions get most messed up, and how do they get messed up depending on expression levels of certain genes, and depending on where. In a general sense, certain regions are more mutagenizable than others based on how much repetitive sequences they have and whether or not they're being actively expressed or not. So
I really like the idea that the packaging of DNA, the methylation, histone wrapping, all that stuff. I wonder if that provides protection. Maybe that's part of why cells do it, and wrap up all this stuff, is to protect themselves from mutations, which limits the mutations, like you said, to the ones that are actually unmethylated, not wrapped around histones, just kind of out there in the open. And so it puts them at risk, but they're allowed to express during that time. So it's kind of like take a risk to express and then wrap it up real quick.
Yeah, like in another extreme, right? So that, I forget the name of the microbe, the one that's extremely radiation resistant. You can basically shred its DNA using gamma rays, and it still chugs along. I think its DNA is like, it's configured. It has enzymes that do repair to double-stranded breaks that are basically kind of already preloaded all over its its genomic DNA. So essentially, if they get to work, if it gets shredded, there's already a team of enzymes ready to patch it up. So and of course it evolved under these harsh conditions to basically withstand that type of damage. So there's a lot of variation. I would assume in DNA regions, and also cell types, based on how the DNA is packaged and so on and so forth, that would affect which areas are much more capable of being mutagenized. So it is stochastic, but like within constraints, within particular.
It's a very good point. I had one other comment on the paper. You kind of skipped over this part, but they mentioned that procollagen was also upregulated in these cells that they delivered it. And that surprised me, because the only thing I think of that would cause that, I don't remember which section it was in. Okay. Sorry.
Just leave this up here.
The only thing I can think of that would cause that is if the presence of the collagen mRNA, or collagen mRNA, triggered the cell to produce more procollagen to then get that to express into the actual collagen proteins. Or they accidentally got some procollagen out of the source cells along with the collagen. Like they didn't mention how they got how the procollagen came to be. They just mentioned that they did some assays and they found that procollagen 1 was also upregulated significantly. Anyone have any other ideas on like how you'd end up with that? Procollagen 1 being the precursor to collagen, which, if I understand correctly, I might be very wrong on this, so someone please correct me. You have the collagen 1A1 gene, but in order for it to express, I think you need procollagen present, and that leads to it.
Like so I got to pull up a figure for collagen production, but I think, let's see. Well, collagen is actually collagen itself, the actual collagen that exists outside of the cells. It's built up of, so you've got your little pieces of collagen, and then they're assembled outside of the cell. I don't know exactly how, but basically sort of like building blocks, right? So sort of so those little sections act as monomers for fibrillar collagen, because collagen is too long to fit inside of a cell, like the actual final version. So it's secreted out in these short sections. And I think maybe that's what they're referring to as those short sections. Well, let me see. Let me find the section that you were.
I just found it as before Figure 2. Actually, the paragraph after figure 1.
Paragraph after figure.
Yeah, sorry, I'm on the HTML version, but it should be around there.
Oops, let me just move this
pretty early on.
toolbar here. Oops. Let me just pro.
You sort pro dash collagen.
Here, pro dash collagen. Here we go.
And there you go. And that's the only time I mentioned it. Just like that one sentence.
Yeah, I gotta. It could just very well be that yeah, I've got to take a look at my old slides on collagen production. It could just well be that basically that the col1 protein is basically the matured version of that variant of collagen, and procollagen 1 might be what's actually being encoded by the col1A1, but don't quote me on that. I might be I might be completely wrong.
Sure. But speculation might be a reason, is that col1A1 may actually encode procollagen and possibly something else, and so that's what's being expressed, and then col1 is then downstream of that.
So let me actually let's see. A slide somewhere on this, but sometimes you can just find this easily here. Yeah. Oops. Yeah. Okay. So let's take a look at this picture here. All right. Screen share. So I think you can see my screen here. So you get, procollagen is what's actually, I think, encoded by the col1A. So it basically forms this short triple helix, and then it gets secreted, through the Golgi, and then it gets assembled in the extracellular space into, the mature collagen, which is way longer than the cell. So because otherwise it would just burst through the cell. It's too long. So so I believe what's being encoded is the actual procollagen, which is, the whatever it is on that 4,000 nucleotide mRNA, and then it gets wrapped into this its triple helical form and gets modified, hydroxylated, so on and so forth, I think, and then gets released, secreted, and then there's probably some additional enzymatic activity out here, which basically cross-links and forms your giant collagen fibrils.
Gotcha. So procollagen is just the one strand, and the final thing we call collagen is actually a bunch of those all woven together into a yarn. Yeah, and there's different types. There's like collagen 1, 2, 3, 4, 5, 6, 7, and depending on where it's expressed, so they're all depending on how it's kind of linked up outside of the cell. For the ECM, for example, I think it's like collagen 4, which is linked up in these shorter, like sort of forms that form like a meshwork, and the collagen that's found in your tendons, for example, is like much more longer and fibrillar, because that's the role it plays. But yeah, so that is it. So I think that's what they're referring to. All right, so very cool. First step forward to get rid of wrinkles, but as we saw from this technology here, not quite ready for prime time, or maybe it is. I don't know. I mean,
I could see somebody using those kind of dermal patches to get rid of crow's feet on their face, right? Put that there.
I think this is superior to existing like collagen by injection solutions. I think like traditional.
That's true.
Cosmetic surgery.
That and certainly superior to injecting a botulinum toxin into your face,
yes.
which is currently medically approved, right? So
Yep.
we can still say how long does the mRNA here last? If we're going to get an allergic response from these EVs, but people are still injecting Botox in their faces, right? And with needles and which basically causing your nerves to basically stop functioning, and not
Bar is low.
Yeah. So I think an HA dissolvable microdermal needle patch with EVs loaded with collagen mRNA that's all basically human components. It's probably going to be safer. And
I'd like to see them switch over to DNA, so we get. I mean, given those effects, and if we're assuming that collagen production is somehow damaged in the cell, either due to aging or UV exposure or whatever, I feel like DNA is the more long-term solution. So you get more than a month of expression. You get years, hopefully.
Yeah, but the good thing about this is that it lowers the bar for acceptance, right? Because I'm sure you're going to need more. I mean. I would definitely see the mRNA, especially given that the whole mRNA delivery systems for vaccine delivery were widely adopted, albeit through emergency use authorization. This bodes well for acceptance of this technology using mRNA. So DNA. Perhaps a much bigger hurdle to overcome. You want to make sure it doesn't get integrated into wrong spots, for example, and cause tumor growth and all that stuff. But if you just have EVs that are mainly packaging transient mRNA that's only expressing collagen or maybe other things. I mean, certainly I think it's a great first start. I think with they might if you're getting this kind of robust transient response with just collagen, there might be additional factors that basically could be screened using these extracellular vesicles, right? So I guess one next approach could be, well, I mean, we discussed there's some effects happening just from the unloaded EVs. So there's a possibility of a whole bunch of other bioactive components there that can just focusing on the wrinkling, right? I mean, there's probably other beneficial effects that are anti-aging, but just from the wrinkling perspective, there's probably a whole bunch of other molecules that because extracellular matrix is not just collagen, but there's fibronectin, there's a whole bunch of other stuff in there. There's elastin is another major extracellular matrix protein, as the name suggests. It contributes to skin elasticity, right? So there's a bunch of other stuff that they could throw in there and see whether or not you get a really turbocharged anti-wrinkle response. And I would expect that to be the case if they threw in elastin and as well as collagen and perhaps some other ECM components as well. Into the EVs, which probably would be a next series of experiments.
Yep.
All right. Well, that is all I have, folks. I think the future is looking bright, at least for transient wrinkle alleviation, if not outright immortality. We'll get to that later. So I don't know if anybody has any other further questions on this paper.
Yeah, when can we get it?
When
can we get it? So I don't have to look at all these wrinkles anymore.
You know what? You could we've got everything out here laid out. You could certainly start your your biohacker lab in the basement and make your own EV system with microdermal needles and and put on a whole like microdermal face mask and see what happens.
Okay. Well, yeah, it'd be nice to actually be on the camera for once and not have to use a filter. I think most people at home can relate to that, right? So it's going to be worse now because I've just bought a new camera. So I'm not using it now. So I've just bought a lovely streaming camera.
Mm-hmm.
So I'll be coming at you in proper 1080p, not this cheap $20 1080p.
You got the camera, but you also need the collagen treatment, right? Before you actually do the 1080p.
Yeah, I'll just use a massive. I'll just filter it.
People are just gonna live like if we don't solve the aging problem in real life, people are just gonna live online and have like these filters and these AI systems just make them look perpetually young and sound young, and everybody's just will be living online. Just it won't be able to tolerate themselves out in the real world.
Well,
That's right.
that kind of thing is already sort of happening right now, and we're seeing it in movies and things. We did an article not long ago, which was based on the new Indiana Jones film, where they're going to de-age Harrison Ford.
Yeah.
But the thing is, you can filter all you like, but aging is still there. It's still a problem. So
Yeah.
there is that. But yeah, it's looking promising. There's a lot of this sort of technology coming along now. I think COVID's actually really helped sort of bring these delivery systems to the forefront, and that's great. So good news for our field in particular, and yeah, I look forward to that. I mean, who doesn't want sort of plumper, more elastic skin that doesn't wrinkle, and bounces back after you finish frowning?
Yeah. Well, I mean, and sure, I mean, we all want to look good, right? But appearances aside, all of these components, collagen, elastin, so on and so forth, are there for skin health. You want your skin to be resilient and less able to tear and get damaged when you smack into things. And all those components are necessary for skin integrity, which is one of your largest organs of your body, and basically keeps out all the pathogens. So I mean, vanity aside, you need all that stuff just to be healthy, right? So I mean, yes, you'll end up looking good as a consequence, but I mean, that's also a matter of health.
Yeah, I think looking good is the side effect of being healthy.
Yeah, but it's also the thing that will actually capture public attention and motivate more people to support us. And of course, skin is very easy to work with.
Mm-hmm.
It's very accessible. It's not like internal organs that are more problematic when you're trying to prove rejuvenation. So it's cool, right? And of course, with the collagen production also is going to affect your arteries and things like that, other parts of your body that use collagen. So I could also see some potential benefits there regards things like hypertension. If you can restore elasticity to the arteries, again, that's also a win-win. And well, I mean, collagen is used in so many different tissues and connective tissues in particular. So it's got pretty broad application, something like this. And so, I'm going to be cautiously optimistic that we may see this, and it may also inspire more support for the field, which is exactly what we want, right? Because we want people to take us seriously and, understand that this is not woo or pseudoscience, but this is actually a very real thing. So
yeah,
win-win. Let's start with the skin and take it from there. We've all got skin in the game, after all.
Har. And if you want to hear more about it live, we've got when is our conference this year? It's a mixed hybrid, isn't it? Live action and online.
Live action. It's not a film, Oliver. It's not. This is not a manga. You always ask me this, and I'm never sort of.
It's later. It's usually later in the summer. End of the summer.
It's always that sort of time. It is
right before fall.
August the 10th and 12th, which is nice. It's about the same time as it was in previous years.
Right.
It will potentially be, I say potentially, we're aiming to do it in New York City.
Yeah.
I can't say too much though, because if I say something wrong, Christy will crucify me.
Yeah.
We are trying to make it a hybrid event.
Yeah.
So hopefully then there's going to be the option of obviously attending the physical events. In previous years, they've been very good in New York, but for everybody who can't make it, we're hoping to have an online element like the previous, like the COVID years that we've had to do a virtual conference, and they've been pretty good as well. So we're basically trying to have our cake and eat it this year, and so more news on that soonish. We are starting to get things in place. So yeah, we should have something to announce maybe in the next month or so, and we can then start obviously talking about who's coming and what's going to be on. So anyway, we will keep you posted on that one, Oliver. Well, you've got to be there because you're the MC. So
yeah. Anyway, hopefully everything is, knock on wood, we don't have another pandemic or something else ugly rears its head. Things have kind of got, I mentioned that I was at a trip to Canada just recently, and I wasn't able to go there for a number of years because of the pandemic. They had the borders closed, and I was pleasantly surprised to find that when I arrived at the Montreal airport and left Montreal airport, it was almost as if the pandemic never happened. It was very, everything seemed not quite, but almost back completely to normal. So I'm hoping that, that we're out of this madness and, can move on with everything that we've got on our plates.
It's been pretty normal.
Travel-wise. Yeah, certainly travel-wise, yeah.
Although I did go on holiday and we got COVID. Whoops. That was last year. It's all right. It was on the last day, so it didn't spoil it too much. But let's just say it wasn't exactly fun traveling when you were like ill.
Yeah.
So it wasn't that bad, in all honesty. The one day was a bit bad, but then after that it wasn't too bad. And I, as I said, like a couple of years ago when it all started happening, I said if it sort of obeys its evolutionary drives anyway, the virus will tend to mutate and become more benign, so to facilitate its infectiousness, but without killing the host. Yeah. So it's not a good strategy for survival if you kill the host, right? Yeah. And it does seem to have appeared to have happened, but it does, as I say, the positive side, as I mentioned earlier, is sRNA technologies. They've always been around for many years, but I honestly think that the pandemic's brought them to the forefront. I mean, case in point, this paper today uses technology that's similar to the vaccines, and I think we're going to start seeing more of that. I recall seeing that some people are going to try and use Yamanaka factors delivered in a similar way. So honestly, it's a terrible situation, but some good has come from it, and quite a lot of progress in the delivery systems of vaccines and other therapies. And it could be win-win for airfield in particular. And of course, as the pandemic has shown, it is quite it's a technology that's able to be sort of built at scale, which is super important for airfield and therapies that we're developing, because we want everybody to have access, right? It's no good developing something that works, but it's so prohibitively expensive, it's impossible to make it practical. So sRNAs and similar delivery systems, I think are one of the things that's going to pay dividends for us in the future in airfield. But there you go. There's some musings from me about delivery systems, and there we are. They're a little bit of an interest of mine. So, but there you go. That kind of makes sense, doesn't it?
It does indeed.
Seeing as what I do for a job. Right. Well, we
All right.
will see you all next time. Thanks to all the heroes who joined us, and who the people out there in Facebook land, etc., who are watching or watch us later. Hopefully you found this interesting, and thanks to the heroes for supporting us to make these shows possible. And if you'd like to find out more about how to become a hero, a lifespan hero I should say, if you visit our website at lifespan.io/hero or heroes, they both work. And as I say, we will see you next time, and hopefully we might have some more news about the conference.
Yep.
So until next time.
Take care.